Hello everyone. So in this video we are going to focus on cultivation of viruses and also on enumeration of viruses. How can we compare viral loads? So let's start with cultivation. So how can we grow an animal virus? If it is an animal virus, we have one of three main approaches. Number one, to use an intact animal. So we will inject this virus into an intact animal and the virus will grow and multiply inside the animal. This could be, for example, a rabbit, a guinea pig, or a mice. As you can see, these are small animals that are easier to handle and to maintain at the space of the lab. Alternatively, we can use embryonated eggs for this purpose, and as you can see here in this image, we have different layers and different tissues within the embryonated eggs which could be injected with the virus. And the third approach is to use a tissue culture. So that is an approach that does not need a living organism. We are doing this in vitro. It is not an in vivo approach. So what we do, we grow cells in a culture media like that, and we cultivate the virus in these cells. So how do we prepare this tissue culture? We start with a tissue. As you know, this tissue represent the specific range for that virus, so the specific host range of that virus. So let's imagine a virus infecting liver cells. So we will get a tissue of the liver, and then we cut the tissue into small fragments treated with a protease enzyme that help us to fragment or disrupt the tissues that join these cells. Once we have the cells, we disperse the cells on top of a nutrient media to support the growth of the cells. So now we have a confluent monolayer of cells through which the virus will be cultivated. Okay, so which one of the three approaches do you think is the best? So this one, the intact animal, as you can see, there is a possibility that if you are interested in a virus that causes a severe disease, it may lead to the death of the animal at the end. And obviously, this is not preferable. Then the embryonated egg will be slightly better approach because you are not causing the death of any animals, but still both approaches suffer from the fact of using needles and injections. And again, there are the hazards of mistakes during injection that the researchers may, by mistake, get injected with the virus while injecting the eggs. So for that reason, the best approach is the tissue culture. It doesn't involve injecting the virus into living cell. There is no death of the animal. Here also, a bite of the animal can transmit the disease to the workers and things like that. We avoid all of these side effects or all of these pitfalls when moving to that tissue culture. And since you can get a specific tissue, you can study more specifically how the virus grow in this specific type of tissue. Keep in mind that we have also bacterial viruses, bacteriophages. So in this case, we will grow them on a layer of bacterial cells. So very thin layer of bacteria. We call it a lawn of bacteria. And once the virus is specific to that bacteria, if it is a lytic virus, as we will see later on, so lytic viruses will cause the lysis of the bacteria, and the areas where the virus is there and growing will produce what we call plaques. So this plaques, as you can see, like dark areas, are actually areas that the virus caused the death of bacterial cell layer, and because of that, around the virus, the area for the growth of the virus, we got this empty holes, which represents the death of the bacteria because of the growth of the virus. Okay, as we said, this will happen in the lytic cycle, because actually viruses, when they multiply inside the bacteria, there are two types of cycles. Some viruses, they do what we call the lytic cycle. Other viruses do what we call the lysogenic cycle. So let's start with the lytic cycle. So what will happen? Both cycles start with the same two steps. So what will happen? The phage will land on the bacterial cell. The phage will inject its genetic material, as you can see here in number 2, inside the bacterial cell. Then, if this virus can induce the lytic cycle, we call these viruses virulent phages. So if it is a virulent phage, it will induce the lytic cycle. So what will happen? The virus, as you can see, while injecting this genetic material, the chromosome of the bacteria will lyse into pieces, small pieces like that. So the chromosome, which is here shown in green, will lyse into small pieces like that, and then the virus genetic material will get replicated. So we get many copies of the virus genetic material shown here in purple. Then the assembly of the virus particles will happen. So this genetic material will be packaged in a capsid, and maybe we have the different particles like that. So, and then there is no way for this particles to exit except after the lysis of the bacterial cell. So since this cycle ends with the lysis of the bacterial cell, we call them lytic cycle, meaning that they end up with the damage of the bacterial cell, its bursting, and the release of the virus particles, or the virion, as you remember. Okay. Alternatively, some other viruses, they are not virulent. They are temperate phages. So they are like mild phages. They are temperate. They are not virulent. They will not induce the lytic cycle. So what they will do, they will go through the lysogenic cycle. So what will happen? The virus genetic material will integrate into the chromosome of the bacteria. And after the virus material has been integrated with the chromosome of the bacteria, we get what we call the prophage. So a prophage is a term that we use for a viral genetic material that has been integrated into the chromosome, and this happens with temperate viruses that go through the lysogenic cycle. So what will happen next? So in step 4, as you can see, this bacterial cell will divide into 2, multiply into 2 by binary fission, and each one of the daughter cells will get a copy of the chromosome plus the integrated prophage. This could continue forever, or under some certain conditions, the prophage can be excised and the chromosome gets circularized again without the prophage. So now the prophage has been separated from the chromosome, and under some conditions this may start the lytic cycle. So there is a possibility that a temperate virus which started with this lysogenic cycle, under certain conditions it can also cause the lytic cycle to start. So that's why the two cycles are represented connected like that, because there is a possibility of moving from this lysogenic into the lytic cycle. So this slide summarizes the two types of cycles that may happen when a bacteriophage invades a bacterial cell. So I will leave it for you to go through it, and as I said, it summarizes what we have already discussed.